So Thomas is going to introduce Sanjiv, and then we'll move from there.
Okay, welcome everybody. It took a little bit of time and I make it very short. I'm Thomas Müller, and my contribution to this talk is the first slide. Nothing else really, a little bit of collaboration. But it's a pleasure for me to introduce Sanjiv Shrivastava, who is a PhD. He is from Johannesburg, from Witwatersrand University there. He did his PhD in physics. He teaches in physics. He organizes the courses at Witwatersrand for the students there in physics, and he has quite a long career in teaching as well as in science and research. We know each other for quite a number of years through to his wife, who is a political scientist here at the Athabasca and an adjunct professor at the U of A. For me, it's really a pleasure to see his talk really developing and his technology. I think we wanted to have a talk on new technology, and I think he's really one of the best to speak on new technologies. And in relation to Singularity, he is known for his mastering and knowledge of the AFM, the Atomic Force Microscopy. I didn't know anything about it beforehand, but I think it has huge value, and at the moment we are in this process of translating his knowledge into biological applications, and we have some data on applying it in nephrology. So Sanjiv, it's really a pleasure, and the podium is yours.
Thank you, Thomas, for the introduction. Well, talking about singularity and technology, the main issue happens to be what do we mean by each one of them. So let's just look at what technology is. A general definition with technology is usage of knowledge and tools in order to produce some gadget or system which allows us to produce a certain function or to enhance a certain function. And as far as technology is concerned, we've been using nature as itself as a simple tool in order to produce, to do a certain function. Philosophical debates of technology have been going on for years. As far as we know, I'm sure even the cavemen were discussing technology at that cave during those times. And here I've got two simple demonstrations of that. 2.5 million years ago, we developed the technology of stone cutting tools using stones. Today, we do the same function of cutting objects except using lasers. At the same time, we can use laser to cool down atoms and probably hold them into a stationary state. With technology itself, it's not just the humans who have been using it, animals use it all the time. I'm sure the first system of cellular organisms that started growing or developing, they were using technology because it's using what is available to them. And here is an example of even the chimpanzee trying to use technology. And in this particular case, it's the humble stick that is being used to gauge how deep the river is so that they can cross and their entire troop can cross through. So that's a bit of the technology. Then comes the issue of singularity. And of course, we can't talk about singularity without invoking philosophical thoughts. Philosophical thoughts have been going on for a long time. Now here I've got two issues that I've represented a bell, which is from the Buddhist temples in Burma. Hopefully now with the new democracy there, we'll be ready to go and visit the place and see the temples and the bells there. This is a bell that was made in about 800 AD, and the structure of it has got a lot to do with the resonance that takes place in each half of the bell. The next slide that I have here is actually on the Big Bang expansion theory. If you look at the part where the brightest on the left-hand side of it, that is the point where the singularity actually took place at time t equal to naught. And that is why we often say we can never have negative time, because every reference thing that we are dealing with is at the start of the Big Bang. For the first few seconds since the explosion, there was a lot of particles that were being evolved, and as they kept on interacting through nuclear processes, you had formation of larger galaxies, stars, and the rest. So this is just a slide that looks at the other part of it. At the time of 10 to minus 43 seconds, what is zero time? We don't know. We're trying to see at this point in time, as the theories stand, we have time t equal to 0 is 10 to minus 43 seconds, which is a Planck's time, where you had lots of particles or gluons and photons that were involved. As time progressed, within a few seconds or fractions of seconds, you had photons being produced, photons interacting with one another, forming quarks, quarks forming antiquarks. They were again going through a different collision process, producing protons, neutrons, electrons, neutrinos, and the rest, and the cycle goes on and on from there. Now, the significance of this is if we want to address singularity, we need to look at what is the basic concept of singularity and where does it come from and what does it involve. If you look at the diagram on the left-hand side and the center two columns that are present, the one deals with unified forces, and your forces start evolving into the strong gravitational force. You have got electromagnetic forces, strong nuclear forces that are involved in it, and the weak interaction. So we need to, if we want to harness singularity, we need to understand these particular forces. In order to understand these forces, we have to look at what the elementary particles is. What is it made up of? And in order to do that, we go through different processes of looking at structures in terms of photons, gluons, Z particles, or bosons, and sigma particles, which all combine. That is the table that you have on the right-hand side, which lists the different types of particles, elementary particles that we have, called the quarks, leptons, and bosons. Quarks is basically light, leptons are trying to make the matter, and bosons are combining both the forces that are involved. The table on the left-hand side gives you a distinction between what we classify as matter and forces. To exploit singularity, it's not just good enough to exploit matter, we need to exploit forces as well. Going further from that, in order to actually observe the particles on the elementary particles, we know the size of the universe is huge, but in order to see these minute particles, we need structures that are available to us. This is at CERN, Center for European Research in Nuclear Physics in Switzerland. The red ring that you see on the top image is 27 kilometers in in length. And the fundamental particles that is where we observe them. But that's not it. If you look at what the actual detection system is, those structures are as high as this building that we are dealing with, but it is at least 500 meters underground in order to observe those structures. With that comes a much interesting point where we were able to see different, this is called the Hadron Collider. Now in here, right there you have people standing that would give you an idea of what the size of this structure is. And the actual process would take place somewhere in the center of this, way deep inside this particular Hadron Collider detector. The recent experiment that is going on for finding the Higgs boson is the ATLAS project. And the dimension of the detector is about 44 meters long and 25 meters in diameter. So we need huge machine in order to see the minutest particle possible. This is fine from one aspect of understanding singularity. However, we cannot use that for our singularity of machines and the brain. Something shifted there. Now, if we look at this particular diagram again, still remaining on the singularity from the Big Bang, what you notice is there is a bar on this scale here, which has got three concepts that we are interested in. One is time, temperature, and energy. Now, in the first diagram that I had about the elementary particles, we often had, when we're talking about particles, instead of talking about their mass, we talk about the energy, rest mass energy of it. Just like what you would do with DNA, instead of talking about its size, you say the number of atoms or daltons as the unit that we use for it. So we start now exploiting this interaction within the nucleus and the atom, and then we can move forward. Now, just to go back a bit, it took almost 400,000 years in order to get the first 8 elementary particles or elements in the periodic table. From there, it took us at least a few million years before we could get all the heavy elements that are involved. But we are trying to shrink all that into a space of 50 years of research. Now, so that was fine with the elementary particles. Now we move on to the atomic scale. This is a transmission electron microscope. This one here is the earliest version of an electron microscope, and Rushka actually got the Nobel Prize for it in 1986. Now we have the latest transmission electron microscope at the Lawrence Berkeley National Lab, and it has the resolution where it can see not only the atoms, it can even try and map out the electronic cloud around the atom. And this image here, although not visible on the screen, these dots that you see here are actually single gold atoms. And this is a grain boundary, so the arrow here, if unfortunately because of the light, you would see that there was a different arrangement of atoms that was involved. From being able to see the atoms and the formation of the Earth and the Moon due to explosions and reaction taking place, going to the cellular stage of single cells being developed about 3 million years ago, and looking at major cell structures being evolving through. Now we're trying to work backward. So if we want to reach singularity, we need to start going at this, we need to start manipulating objects at single atomic level in free space and free position. That is when we would be reaching singularity. Manipulating an individual atom is very easy. We can do it on a substrate, but that has to be very large. As far as singularity of the human is concerned, well, I've just tried to look at some technical drawing of wiring of a human being with all the circuits in it. That is relatively easy. The image that you see here, which might look like firing taking place in the brain, is actually the activity of the internet that is taking place. And if you think of the processing speeds that are taking place within this web of signals, it's faster than what's happening in the human body. The neuron firing is of the order of milliseconds. The information you're getting down is of the order of milliseconds to gigahertz that is involved. In order to start manipulating the atoms, there is the concept of channeling that's involved. That is, this is arrangement of atoms. The round blobs that you see are arrangement of atoms, and you can fire a single atom through that, and we can start manipulating what's going on with that structure. The same going into your grain boundaries. And in order to do that, nowadays it has become much more easier to deal with an atomic force microscope, which allows you to manipulate atoms one at a time. Now, if you see what is relevant here is the shrinkage of the technology, which yet we are now able to start manipulating within its scale. So now we go on to the second part of it, which is nanoimaging, which hopefully would be of interest to most of you. This is a technique based on a very simple principle which all of us are now using without fail. Everybody has a computer which has a hard disk in it, and it's working on that principle. A simple movement of the record player. That is what an AFM is. Let's see what the technique is. So I'll try and describe what the technique is, what it can do, and how we exploit different aspects of it, and we can use it for the study. What you see here, if you take a few seconds to look at it, what looks like just round blobs all over the place, bright spots sitting around, are actually atoms. And this image was taken from something like that. So you have individual atoms that are present here. Those are the round blobs. This was an image taken from a structure that looked something like that, where you can see flakes and the rest that are present onto it. And if you try and decrease the magnification of that, you see some sort of lines that are present on there. These are straight lines as if the thing was actually cut, which you further zoom out of that, all this information belongs to the most humblest technology that we have, and which is the pencil. So all the images of the atoms that we were looking at were actually evolved from this simple pencil of graphite. In terms of the scales that you're looking at, this scale bar here is about 90 microns in size. The image size is a few nanomicrons as well. Now I've got, as soon as we go into digitization, what is important is the size of the pixel. If you zoom out of the image, the last pixel that you get before distortion starts taking place, that would determine the limit of your imaging capabilities and the information that one can get. So whenever we talk of imaging, there are two types of limitation we have. We have limitation due to the technique, and there is limitation due to the digitization of it. This is an optical image of the pencil being sharpened at 300 magnification. That is the image in scanning electron microscope at 90,000 magnification, where you see the flakes that are present, and that is the STM image where you can now resolve the atoms. In terms of the resolution, think of it as if you're sitting on the moon and being able to see the concrete grains in the building from outside. So that's the depth of resolution that we are looking at. What is involved in order to do that? These are the two main people who were involved in designing and developing the technique called scanning tunneling microscope, the predecessor to atomic force microscope. This was the first STM that was built. With that, they were able to observe this ring of atoms. These are silicon atoms in a certain orientation. So the little blobs that you see are actually silicon atoms that are present. This image here are actually individual gold atoms. Now, in order to be able to see this, there are two ways we could look at it. We could either look at it in terms of electronic signals, or we can discuss the whole issue in terms of forces. I'll spend more time looking at forces rather than electric current. So the force level that we are dealing with, or the energy levels, are of the order of the bond energy holding two atoms together. So it's 10 to the 19 joules. Bond length is the spacing between atoms, which is of the order of few angstroms. And the typical forces are 1 to 3 nanometers. Now the relevance of this becomes important when you start later on looking at what are the forces that are holding the DNA or the proteins together. We need to know this kind of information. In terms of the bond energy that is involved, we can look at it from a quantum mechanical point of view, or we could just simply stay to the typical force in the macro scale. We could also look at Coulomb interaction. So if you're interested in looking at whether the arrangement that you have is ionic or not, and what is the value of it, you can look at the Coulomb interaction into that. The basic principle of atomic force microscope is within the weak interaction forces, there is something called the van der Waals forces. And this is a typical graph of a van der Waals forces, that if you have two atoms, one sitting at this point, one coming further away, as the atom starts coming closer and closer to the one that is stationary, at some point there will be attractive force that is involved, and after a certain point you have a repulsive force that comes into play, but the two will never meet each other. You will not get a scenario where you get the two fusing together of the atoms. You can have fusing of nucleuses taking place, but on its own with the atoms, they will observe this particular van der Waals forces. This part is the repulsive, that is the attractive forces. The attractive forces could be your Coulomb force, and there's several parameters that are involved in it. Could be your magnetic as well that comes into play. The basic mode of operation of the instrument is in either exploiting the repulsive force information or exploiting the attractive force information. If you're going to exploit the repulsive force, we end up calling it as the contact mode, and if we exploit the attractive force, we call it as the non-contact. This is just terms that are frequently in use, so I thought I would introduce it right away rather than bringing it up later. The basic idea is if you have your structure that is stationary, the probe that is moving through, this is the tip that is involved here, so between your atom there and the stationary atom, as it goes through, it will measure what is the interactive forces. So either you could have your sample that is a stationary, so your cell that you're interested in could be stationary, or you could have the tip stationary and your sample moving under that. Now, one of the problems with atomic force microscope is your sample or the feature of interest has to be fixed. It has to be attached to a surface in order to observe it. We observe it using probes, so very much like the needle that you have on the old-time record players or in your hard disk. There are different shapes of it as well. What we can also do is functionalize this and add different type of elements on it or proteins in order to use the measurement. So the basic idea that we are dealing with is the force and displacement are related, and this constant of its relationship will give us information about the surface that we are trying to observe. At the same time, you can oscillate this structure, bringing in resonance structures into it, which will be of use. Now, with any technique, there are different aspects of it. So with AFM, the 4 stages are scanning, the scanner on which you place the sample or which is used to scan the image object would be of use. Then you have probes and tips that I showed you an example of that. And of course, electronics, feedback, and image processing. Whenever you're acquiring digital information, the reality is all the information that you get is actually a point information, and then we recombine that to generate a 3-dimensional image, or these are all pseudo coloring that is involved. It's the same structure, but different coloring doesn't mean that actually your structure is anything different. So we have pseudo coloring that is involved. The reality is everything is your single pixel. For any electronic based system of observation, we have to have a feedback loop that tells it what it should do next, or to preempt what the next feature would look like. So that's your feedback system that we have with a huge number of parameters. And I think the animation here explains the scenario very well. If your parameters are not properly optimized, then you do not sample your surface. And if your parameters are very large, then you actually get damaging of either the surface or you get damaging of the tip that is involved. And in either of the cases, you would not get the information. So feedback, having it properly optimized, is an essential tool to it. Now, because I'm trying to combine things to singularity and we're interested in resonance, that is how we know information about from the time of the Big Bang, what we end up doing with the AFM as well is, this is your ordinary watch. Your quartz watch have actually a tuner in it, like very much like for what the musician used to tune their equipment. You have a tuner that is present in there, which has got 2 arms into that. They're made of quartz. One acts as a cantilever, the other one acts as a tip. You can start oscillating them and accessing different type of information into it. So this again goes to, if you have the oscillating system, you can look at the displacement, that the force of interaction between the probe and the sample can be related to some factor of the mass that is involved as well as the displacement. I'll try and avoid going too much in details of it. I don't think it's of most people would be interested in that, or you can read in the literature. That's much easier. So if you've got two interacting particles that are further away, or objects that are further away, as they come closer, you have some strange behavior that is involved in it, either an exponential graph that is involved or some complex numerical format. If you look at, if one is interested in absorption with surfaces, so you can look at either the conductiveness between your probe and your sample, which might be of interest with the biological systems quite a bit, as well as if you're looking at drug delivery systems, you are interested in how much of absorption is involved or how much forces are required in actually creating a separation that would occur. Your vertical force and interaction energy can all be measured using this particular technique. How to actually do the interpretation of the data that you get is the tricky bit into that. So from your force imaging and atomic resolution, you actually can not only see the atoms right at the top, you can also access information from below the structure as well, but it is just atomically below the surface and not far deep into it. Now here I'm going to just show some of the measurements that have been done using this technique, and pardon for not being able to give you more medical details, but hopefully it will give you an idea of how the technique can be exploited in the field. So these are your mRNA released from cell nucleus through nuclear pore complexes, and this is measured with AFM. So you can see that these are nuclear pores before anything was done to that, and after 20 minutes you find that there is a change in the structure that is involved. And if you just zoom into one single structure here or a single nuclear pore, you can see whether it is open or closed. A much better work was done by another group from Basel in Switzerland, where they were able to show your closed and open, were able to identify the pores that are open and closed much more neatly into that. And that's your 20 nanometer of scale. These are your, each pores are of that order of size. You can see there is an atom that is visible in the center of it, and that is your closed system. And then you get here, which is an open system, where you get the ring that is present. Those are each nodules that are visible. You can look at your endothelium cells as well from it. Now, because this technique gives you true three-dimensional information, you can not only just do the mapping of these particular structures, but you can also measure the volume of each of these cells that is involved, and then you can compare it to either their wet or their dry condition. Here you can see that there is a huge change that has occurred. On the y-axis here, things are in thousands, and on the y-axis in the dry case are in hundreds. That tells you that there is a significant change in the size of your endothelium cells over during the measurements into it. Another example is you could do a three-dimensional representation of it to see what might be interacting at the bottom of it, and you can then compare in situ, you can compare what is happening to the cells as well. Now, keeping in mind that your interactive forces are of the order of less than 5 nanometers, they are not actually going to destroy your sample. So you don't need to coat them, unlike the case with scanning electron microscope, where you need to coat your sample, or in transmission electron microscope, where you really need your sample to be very thin. Here we don't worry about that. You can simply look at if it has got a surface that has some activity, you can measure it. You can also do live cell imaging as well using this technique. This is a rat tracheus cell looking in a dynamic stage. This is the first image that is observed. These are both the images that were acquired simultaneously, and as time lapses by, you can see the movement of your muscles that is involved in it. In addition to that, because we are exploiting large number of interactive forces in the van der Waals case, what we can do is take the AFM probe, place it at a point, and wait for its response and see what happens. So you can go image a single cell and identify the points of interest and actually look at your response time at each point, so you see the variation in the responses that takes place. Now, not only can we do that with electric signals, we can do the same information using mechanical information, so you can have change in stiffness of the material or the elasticity of the material changes from one point to other. So you can again do that with this particular technique. Now, when we're dealing with oscillating system like the resonance stuff that I had earlier, you not only get information of just the topography, but you can extract three forms of information from it. One called the change in amplitude, so you can have the actual topography. Two, you can look at the frequency response. This is something that we can use later on to see if at what spots or on the cell do you have the interaction taking place or not. And you can see what is happening below the surface by looking at the phase mode as well, which is far more sensitive than the phase, the phase mode is more sensitive than the frequency and the amplitude. And this is what we try to look at. So this is a capillary from an glomerular in the kidneys. You can see that there are three images simultaneously acquired. This is giving you the frequency information, that is giving you the height information, and that's giving you the phase. You see that you have far more information or structural information being revealed in the phase than in your topography mode. Going further, this is what the optical image of the glomerular cell was. We tried to identify that, and then using an AFM, this is a 100 micron image that is seen. This almost represents what we were trying to look at. That's a typical SEM image of a broken capillary just seen. Now with AFM, you can see far more structures that are present in there, which you can't see with your scanning electron microscope because it's hidden below that, as well as the depth of resolution with SEM is not as good as what you get with the atomic force microscope. And then with high resolution SEM, you can see the fenestrate, the ring structures in there. So here is an AFM image of the fenestrate, but this part here that you see are the pores. This part there is the actual cell membrane that is visible. We can also do the measurement of it. You can try and measure what is the size of the capillary, what is the each. So you can do a line profile of it where you see what's involved, go and do the measurements. You can get height, horizontal distance information, vertical height information, volume information as well that I showed earlier. This is again the same image but presented in a different mode where you actually see as if the capillary, the structure is broken, but it's part of the same system. Again, likewise for the fenestrate. Now, you would also get an additional information as to what is happening from one point of the fenestrate to the other. Unlike with the SEM, you cannot from here determine what is the thickness of this single membrane that's involved. However, if you go with the AFM here, you can see this profile coming down from there to there would give you the thickness of the single fenestrate membrane that is involved. But in order to get those images, it is not as simple as your optical microscope, plug and play, put it under the microscope and you see it. Just because there is the term microscope doesn't make it that simple. It is very time consuming. There is a lot of hunting that's going on. So it's almost looking like looking for a needle in a haystack. At times, if one is lucky, it depends on whether you prayed to the Higgs boson or not. You will either see it immediately or you can spend days before you actually see any structure into it. These are just different images in order to actually see what we were interested in. Again, of the same structure, the capillary and the fenestrate, just given that example of it. Again, going through and now measuring the thickness of it, the diameter of each of the capillaries can be done. You can measure both your length, your width. From this, we can also extract volume information. If you had red blood cells, you can go and measure the volume of the red blood cells and the changes that might occur in it. And you also get cases, because you're using resonance structures, you get information from structures that might be present beneath the surface. So these little bumps that you see here might be something that's present beneath the surface. The whole thing with atomic force microscope and all its technique is it is largely a surface technique, and you're looking at outermost layers, so 0.1 nanometers of the surface is what you're looking at. So what is truly on the surface is what you will observe. This I've already shown. Then we have looked at fibroblasts, so you can see your fibroblast structures. Now you can go through, we went through a different aging process and then tried to see the change in elasticity of the cell, and you find that as time goes by, because their viscosity decreases, their elasticity or the Young modulus of the cells actually increases into it. So we can go and select a point there and do a simple indentation, which is applying a force going into the cell and relaxing the force to see how the response of the cell will be. From there, we can determine what the change in elasticity is. So far, I've largely presented things for imaging purposes. So AFM is a tool for visualizing what is happening to your cell. But that is not all as far as the technique gives. These are the paintings from Escher, so it looks as if it's a single roof. I mean, people working on two different levels, but it's just your color coding that is involved into it. And while you're working at it and you're bored, you can get some artistic patterns as well from your sample. We can also do things as far as variation of electrolyte concentration is concerned. And you can see within the same measurement that is going through, as you change your pH level or you change the concentration of the solution that's in use, you get different responses into that, and you can use that information to see what the behavior of the structure is. You can also look at, this is on the top here, these are your TEM micrographs showing negative stains, and then you get AFM images looking at the same structures. You can see that you get far more structural information with the AFM than you get actually with the TEM in this particular case. There have been a lot more studies that have been going on, and of late there are lots of papers coming out using AFM in the medical field. And here is a myosin V molecule translocating along an actin filament. So if you start off as a function of time, you take the first image that is involved, and you see as the time progresses, this particular part of the filament starts shifting and changing its size through. So these are all measurements that can be done using a simple probe and sample sitting on a scanner. This is a 130 nanometers into 65 nanometer scales, and the z-axis is supposed to be your 30 nanometers. So the color range that you see from the brightest to the darkest is about 30 nanometers. So that's your single actin fiber. Now what we can also look at is imaging of nanoparticles in cells by nanomechanical methods. So what they've done in this particular experiment was they were looking at toxicity with nanoparticles, especially carbon nanotubes that were injected into the cells, or sorry, rats that were allowed to breathe that and see what was going on. So here you have these are embedded particles. So this SWCHN is single-walled carbon nanotubes, hollow tubes that are present within the cell. and you can go on the surface and map what's going on with the cantilever, measuring the phase and the topography information, the material sitting on a piezoelectric. That's your normal AFM structures. And what you see from there is you can start seeing toxicity that is present in the cells. So this part here, the blobs that you see all around, are the carbon nanotubes that were embedded or that were absorbed by the rats and can now be imaged into it. So you see the structures of that nanoparticles from A to D are AFM topography. The last part here where you have got the B and D images, this part here and this image was observed using a slightly different mode of technique, which is scanning near field ultrasonic holograph. So they're actually using the oscillation of the cantilever, measuring what is the feedback or acoustic information to determine what is the structure that was present inside the cell. So as far as the AFM is concerned, yes, there are tons of applications in the medical field that is involved, that hopefully as time goes by, it will become more and more popular. I know that in Europe, they actually have a center for developing all the protocols for using AFM in the medical field. So you can use it for protein unfolding, you can look at energy escapes from it, you can combine it with light microscope, you can look at cell surfaces as well. Molecular recognition microscopy is still developing. We are quite a bit away from there. We can also do single cell force spectroscopy, so you can look at the change in the force in the cells you go through. So where does it lead us to in terms of with all this technology? As far as singularity is concerned, we're going back in terms of the structures. We started off there several millions ago, and with technology spoiling us, we are heading back into the same stage. Now, started off by looking at resonance structures. This is something that I just got recently from IBM. As usual, they are ahead of the field. They are looking at nano MRI, so you can look at imaging that is taking place using the same cantilever system, oscillating cantilever systems, and determine what structures might be present through it. So it's extracting information of your using like the typical MRI, magnetic resonance imaging. We're using magnetic resonance imaging. However, the difference is you don't need large amount of molecules. Now you can look at it with just 20 nucleuses that are present. In order for us to get an MRI of the body, you do need several thousands and billions of molecules in order to get an image. Here, 25 atoms sitting below there will give you the information that you're able to see. The nuclear resonance is not as strong of a signal as a magnetic resonance would be from the electron moving in a circuit that is involved. So going back to singularity, looking at everything with respect to resonance structure, we can now look at resonance using a simple tuning fork resonance system. We can look at more and more features that are involved. And with that, there are obviously challenges in the technique, but I'm not going to spend time going into that. I'll stop at this particular point. So there is a lot of mapping that can be done with it. Thank you very much. Any questions?
So there are so many ways of looking at things now. Is there any way to make a decision about what is the best way to look? I mean, even when you take something as simple as 3D, there are people who are predicting that 3D is the future of everything, and there seems to be also a kind of, in some sectors, a massive failure of interest in 3D. So are we going to end up with 1,000 options of a way to look at things and no way to choose between them? Or are there fairly obvious tactical things that would, dictate that one particular way of looking at something, visualizing something is obviously the way to go?
Visualization is one aspect, and extracting information is another aspect. So it's all dependent on what kind of information we want to extract. Now, of course, we are restricted to the electric, magnetic, and mechanical information. Those are the three forms that we have to depend on. The maximum resolution you can get is if instead of dealing with particle interaction, you actually deal with photon interaction into it. So that means, for example, with an AFM, you are actually looking at a field interaction which has a higher resolution than, like for example, with a transmission electron microscope, you actually need electrons to knock out electrons to get information. Here, you are looking at information based on the electric field that is present around the surface versus another electric field of non-characteristic that is being brought in together. So it gives you a higher resolution. So I think it's all a question of what is the key interest. If it is electrical information, then we need to look at something that gives you maximum electrical based response. If you want to look at, for example, with water molecule, the hydrogen and oxygen sitting in that form, our body selectively decides when to allow the water molecule to pass through the capillaries and when to reject it. If it comes with the hydrogen entering into the capillary, it will reject it. However, if it comes with the oxygen going in, it will allow it to go through because the whole process is through repulsion. So this is an aspect of repulsion, the forces that is very seldom in use. So I would say it's a question of what is of interest.
What about throughput? I mean, to look at the extremes, Michael Woodside was telling us of some nanotech experiments that take years to set up, and the right conditions in, almost absolute zero time temperature, it exists for only a very short time, and then it's over. So the throughput in that sense is extremely small. What about atomic force microscopy? Could you examine many things in a day, or what?
In fact, if you look at the last 15 years, the publications using this particular technique has grown now to almost hundreds of thousands of papers a year. Initially, it began with physics with just about 1,000. In fact, most of the people who designed the STM, their papers was rejected by the Phys review, saying, oh, not important, irrelevant, and the rest, and today it's a major tool. AFM has been in use in low temperature cases, revealing much better substructure information. Mike was correct in saying that we can never get absolute zero. In fact, even the universe's temperature is a few millikelvins. Once we get absolute zero, everything should cease to exist. Everything will be stationary. And that's why they've tried to use lasers to slow down atoms to almost make them stationary in free space. If we could reach that stage and keep it for several hours, and then go with an AFM probe, we would be able to see the structure. However, I know, like at Robert Volkroff, who has this incredible instrument which actually can look at the electron density around an atom, and not just look at it from one point, but from different angles as well. And that's where quantum computing would come into play. So 0 degrees, no, we can't. Can AFM give you more information? Certainly, yes. It might be a biased approach on my side that because I'm familiar with the technique and I use it frequently, but looking at what it is being able to generate, and how it has revolutionized the whole technology from MEMS to nanotechnology is because of atomic force microscopy.
What are the costs? Are the costs of the apparatus such that only a few places in the world can have it? Is it within the capability of every university to have one?
It can range from pocket money for some of the rich kids of $500, you can make an STM for yourself, right, to almost $1 million. Okay. But at U of A, there are at least 20 such instruments that are present. Although they are not that widely, people don't know about it. It took me about 3 years to find out that there are so many instruments on campus. Yes. So everybody sits with their machines. Around the world, right now, there are over 100,000 AFMs. And in 1990s, there were about 3 companies manufacturing it. Right now, there are almost 120 companies. So it's become a tool. If you have nanotechnology, or if you have anything to do with cellular level imaging, AFM has become a part and parcel of it. It's a microscope.
Right, I see. Okay, well, I should let others ask some questions.
Thank you very much, Sanjiv. That was really beautiful and overwhelming. My question is, when is it too much? When do we see too much granularity? Or is it that we are not used and we have to learn to see the granularity? If I look at a picture or so, I would see the beauty of a Mona Lisa in the picture, but if I would look at it with an AFM, I wouldn't see the beauty.
There is actually an image of a new Mona Lisa that has been found using AFM, except they did thermal imaging of it. So below the paint, they could see what the actual Mona Lisa looked like. So that's the extent to the technique that is involved. Although the technique is able to give a lot more information, because it is an instrument and it is dealing with the complex number of forces and instrumentation, there are lots of artifacts as well. Currently, there are almost 70 odd different types of artifacts that are present with the instrument. So one has to know what you're looking at. So a prior knowledge helps in order determining what's going on. So I think one can see far more than what you observe with optical or scanning electron microscope. But one has to be aware of how much is an artifact, and those are features that with experience one would know that that's not a real feature. For example, if you see on a smooth, what is supposedly a smooth surface, you actually all of a sudden see triangular structures sitting on top of that. Now certainly no system, organic system, would be made of triangles, because the surface energy to keep the triangle is far greater than keeping a smooth surface. So you know that that's an artifact that is involved. But there are different ways in trying to verify whether it actually was a structure or not.
So you think we have to learn to see what we see, or do we need artificial intelligence to see it? When I look at an electron microscope, there I still can see the original structure. It brings me down and I see the cell. But if I look at an AFM, and we struggle with this, we don't know what we see when we look at a slide from a cell culture or from a nephron or from an endothelial cell.
The main issue is that with optical and SEM, the information you're getting is subsurface information. And here you now start getting surface information at the monolayer only. The whole process is, like you have often mentioned, go the Google route. Start off with a large scan and then start identifying what is familiar that we can recognize, and then eventually go zone down to what is of interest, or in the other case, work with isolated cells, and that might be much easier to deal with. So I think there is a lot of prior knowledge that is necessary, and a lot of patience and hunting around in these images, in the cells.
Perhaps a very basic question, but it seems to me when we use things like optical types of microscopes, we have to prepare a slide, so the tissue has to have been extracted from something. It won't be alive anymore, probably because it was extracted from something. So these kinds of technologies, are they things that have to be performed on tissues that are no longer alive or can't be alive, or are they able to be performed on creatures that might still be alive?
In this particular technique, your cells can be alive when you're imaging them. The only condition is they have to be fixed to a certain substrate. They shouldn't be wandering around because there is already a scanner that is a probe that is moving on top. So you at least want the cell to be fixed at a point, and then you can look at its activity. So you can, for example, in this particular case, this was all the solutions that were flowing through the cell system while the measurements were being taken. So yes, you can do in situ measurements with it. Live cells can be imaged. If you were to introduce, if you wanted to see the response of any of these under electric condition or under drugs, you could apply the drugs to it and see how the cell changes shape. So that can be done.
Okay, thank you.
Welcome.
Okay, well, I think that's probably it. So thank you for a very interesting presentation. Thank you very much.
Thank you.